Welcome to Signals and Synapses, the podcast of the Max Planck Institute for Biological Cybernetics in Tübingen, Germany. I'm Sophia Jans, and today I'm happy to welcome Svenja Brodt and Simon Faßnacht to our podcast studio. Svenja heads the research group Brain States for Plasticity here at the MPI for Biological Cybernetics. She and her team are interested in understanding how the human brain encodes and stores information. Welcome to this episode, Svenja.
Hi, happy to be here.
Simon is a PhD student in the Cognitive Neuroscience and Neurotechnology group at our MPI. He will be co-hosting this episode and talk to Svenja about memory formation. How do we create long-term memories? What role does sleep play in this process? And what does this imply for studying, sleeping, aging? Thank you for joining this podcast episode, Simon.
Hello, thank you for having me.
Over the years, many scientists have wondered why we remember some events or facts for our entire lives while forgetting others within moments. Svenja, you have done extensive research on how long-term memories are formed. But before we delve into your own ideas, could you explain the traditional view of memory storage?
Yes, sure, I can do that. So, we know that memories are stored in the brain, and they are stored by forming new connections between different neurons. And for a long time, it was not clear whether there were specific regions in the brain that host memories or whether memories are distributed throughout the whole brain. And actually, there was a very prominent case, the case of Henry Molaison, or better known as HM, where it became clear that there are certain parts of the brain, specifically the hippocampus, that are very important for memory formation and also for memory storage. In this case,
the hippocampus, maybe for our listeners who are not too familiar with the brain, could you quickly explain where that is?
Yes, so the hippocampus is, evolutionarily speaking, a very old conserved brain structure that lies in the middle, like towards the middle of the brain. So if you think of the brain as a ball, basically you have the outside of the brain, that is the neocortex, that contains most of the cell bodies of our brain. And then when you move more into the inside of the brain, there is the subcortex, and these are more old, again, evolutionary speaking, old brain regions. And between basically the cortex and the subcortex lies the hippocampus.
So people realized that the hippocampus is important for memory, you were saying?
Yes, and that was basically because HM suffered from a very severe form of epilepsy that was not treatable with medication. And one practice that was in the 50s and 60s was to remove parts of the brain where it was assumed that basically the basis for this epilepsy was located. And when removing parts of the brain tissue, also both hippocampi of HM were removed. And afterwards he displayed a very severe pattern of amnesia, so he could not form new memories at all. Whenever the doctors would visit him, he would not recognize them, for example. But he was able to remember very old memories. And that led basically to the first formulation of a theory that is called systems consolidation, where in the very first instance, the idea was that the hippocampus is responsible for forming and storing novel memories, whereas very old memories might be transferred to other brain regions, for example, the neocortex.
That's actually a very good way to pick up with my first question. Since you already mentioned the cortex, and it's one of the most recently developed parts of our brains, and it consists of these several lobes, so one of which is the parietal lobe. To our listeners, when you picture a brain, you probably have an image of this walnut-like structure, including bumps and ridges, or gyra and sulci in nerd speak. And if you looked at the brain from a bird's eye perspective, you would find the parietal cortex at the top center of the brain. Your work showed actually that the parietal cortex also can and sometimes form these rapid memory contents. Is this the same kind of rapid memory as is generated by the hippocampus, or could you elaborate on how they are different?
So yes, this is actually a very, very good and complex question. I could probably spend the rest of this podcast episode talking about this. So in general, I just alluded to it, the old idea was that we have memories first developed in the hippocampus, and then in a very, very slow process, this memory is transferred, and that was the original idea, a literal transfer of the same memory from the hippocampus into a different store, and this store was supposed to be the neocortex. And with more and more evidence, it became clear that, like, speaking very broadly, the quality of a memory that is stored in the neocortex is very different from the quality of a memory that is stored in the hippocampus. So when you think of the hippocampal memory system, it is very densely interconnected, these brain regions. All the neurons talk to each other, basically, which gives it a very high capacity of storing detailed information.
Could you elaborate on that?
So the idea is that specifically episodic memories, which are our memories for facts and events, and episodic refers to the fact that they are very detailed, that you can exactly recall in what place, at what time you have met a certain person and what was the conversation, for example. Whereas memory content that is stored in the cortex is supposed to be much coarser, basically saying in this slow transformation process from the hippocampus into the neocortex, you are losing specificity, but you are gaining generalizability.
Okay, So now you basically explained how these two memory systems, the neocortex and the hippocampus, sort of interact with each other, and also part of the traditional view that short-term memory is rapidly generated in the hippocampus and then transferred to the neocortex for long-term storage. But your findings actually challenge that. Could you elaborate on that a little bit?
Yes, sure. So the original assumption is that this transformation from a more hippocampus-dependent to a cortex-dependent memory is a very slow process that is happening mostly during offline periods, such as sleep. The idea is that during sleep, we recapitulate the memories that we have formed during the day, and through this basically continued training from the hippocampus to the cortex, we can establish a memory in the slow learning cortex. If you think that maybe this is the actual feature that the cortex needs to establish a memory trace, or reactivation, the critical feature, then it is conceivable that if you actually induce reactivation also during a wakefulness state, maybe even active reactivation by re-encoding information or recalling information, that this could be accelerated. And this is
so in everyday terms, that would be if you repeat something that you just learned, right?
Yes. So that is exactly also what we are doing in our experiments. So we invite the participants to the lab, they encode information, and in our case that's mostly associations between different stimuli. For example, you have to remember that a goose always comes along with an apple. And then we ask participants to repeatedly perceive these stimuli, these associations, and we ask them to recall, for example, by presenting the goose and saying with which fruit was that connected, and then they recall the apple. And that is actually something that we all know from everyday knowledge, right? If you repeat information, you will be better at remembering and storing that. The question is just how is that implemented in the brain, and which regions are basically benefiting from this repeated encoding and retrieval.
So what did you find?
When we did that with our participants, and on the one hand, we see in the behavior what we expect. They memorize better. They can memorize for longer terms. So it's a long-term stable memory that we are inducing here. And when we then look at the brain activation by putting people into an MR scanner and looking at the functional activity of different regions, we see that with each repetition, the activity in the parietal cortex that you just mentioned is increasing. So this is per se not the signature of a memory. We have to basically fulfill many more criteria to say we see an actual memory representation in the parietal cortex, but that is what we see in the pattern.
So to summarize, we have these two brain systems that are relevant for memory creation and for retrieval. And you touched upon how they interact. The classical view is that the hippocampus creates fast short-term memory, whereas long-term storage happens in the neocortex. But you also showed that, for example, due to rehearsal, the neocortex is also able to find or to create rapid memory contents. Could you maybe touch upon how you and your group actually research those phenomena? Could you just maybe give us a quick introduction to the projects and the questions you are asking?
Yeah, sure. So if you think of memory, you can actually investigate that on many different levels, and on all of these levels you should see an effect of a memory. So if we start with the behavioral level, if you build a memory, it will make it easier for you to recall the information later. That is something that we can investigate in our tasks when we later on ask people, what have you learned before, basically. And then to actually show where in the brain this memory representation, while I'm recalling the event, is happening, we use fMRI, so functional magnetic resonance imaging, where we look at the activity of certain brain regions while participants are perceiving the stimuli, so encoding the stimuli, or are recalling the information. This activity is in certain points unspecific, so you have to tailor your experiments in order to extract from this activity the specificity to say this is a memory representation. That's why we, on the one hand, add another kind of analysis to just looking at how different regions change their activity levels overall by looking into the patterns of this activity. So with this idea that if there is specific information coded into the activity of the brain, we should be able to extract that from the pattern of the activity within a region.
And are you then able, by looking at the pattern of brain activity, to decode or decipher the content of the memory that the subject was, say, rehearsing?
Yes. So that's basically what we do for this, is to actually not have any visual input, because if we show the stimulus, we never know whether the activity that we evoke comes from an actual memory or from the fact that you are actually also perceiving the stimuli at the time. So basically our participants are seeing nothing, or they may be seeing a cue that leads to the associated memory. And then we are basically recording their brain activation while they are remembering this stimulus. And then we can later on, with the classifier, tell in this moment the person was thinking of a goat, for example, or was thinking of an apple.
That's certainly impressive and a bit scary.
Yeah, like a mind-reading machine.
No. So I think we are like 50 years from that. So that is at the moment, this is very coarse. So we are operating on the level of categories here, right? So we can say this is an animal or this is a vegetable that they were thinking about, but we are not, at the moment at least, not able to very specifically extract what exactly they were remembering.
Okay. Since we talked about functional activation in the context of memory, do you find that functional activation of memory, like the first presentation of a stimulus or the encoding, the rehearsing period or the recall event, are they different from each other in terms of, for example, where they are located and also maybe the temporal dynamics?
So what we see across repetitions is that the activity in certain parts of the brain, and for us that's mostly these higher integrative parts, so medial parietal cortex, for example, activity increases across repetitions, whereas activity in more sensory region, for example, actually shows the opposite pattern. So we have a decrease in activity, for example, in the visual areas that code for the perception of these stimuli. So there's basically a bit of a shift into the contribution of different brain regions. At the same time, we also see that the hippocampal contribution decreases, which basically mimics the effects that you usually would see after natural consolidation, so with this natural transfer over time, over sleep from the hippocampus to the cortex. And the patterns that we see are actually mimicking that within one learning session, so within 1 hour.
So in like complete layman's terms, if I look at an apple like 50 times in a row, you see less and less activity the more often I look at the apple, right?
For the visual regions, yes. And that's kind of, there's different ways how to interpret that. So one part of the field interpret that in a way of just habituation or adaptation. There's just less activity if I have done something many, many times because there's a fatiguing effect, for example. And if you look at it the other way around, any change that happens in relation to the repetition of a stimulus must actually imply that there is some kind of representation or memory that causes the decrease in activity. So you could also see that as a memory signal, which makes the whole story much more complex than it was if you only thought about an increase in activity means higher contribution.
Since you already mentioned, like the actual storage space of memory is difficult to determine with functional imaging because you always sort of are doing something with that memory content. It's not like you put it in a shelf and let it be there. It's like either you are rehearsing it, you are encoding it, or you are retrieving it. But your lab is also trying to look at the storage space. Could you tell us how you are approaching that and whether or not this is sort of different from the findings you get from functional imaging?
Yes. So yeah, the difficulty with the functional activity is that we cannot disentangle whether if we see a memory representation in the cortex that actually means that it is natively stored also in the cortex or whether it might actually be stored in a different region, for example the hippocampus, which then basically just reinstates patterns in the cortex. And this is a super critical question in the field, and to actually try to tackle that with our non-invasive methods that we have to use in the human, we decided to look at brain structure and more specifically brain microstructure to be able to see a change that is related to memory formation in the absence of actually manipulating the memory at the time, so without consciously thinking of it or seeing any kind of stimulation.
So you're looking at whether the person formed new synapses in a certain brain region? Or?
Yes. So I think we have found a way to indirectly look at some factors that co-occur with the formation of new synapses. That's how I would phrase it. So MRI in general is scales apart from the very, very small effects that happen at single or multiple synapses during memory formation. The method that we are using at the moment is called diffusion-weighted imaging. And to put it very simply, this technique tries to look at the movement of water molecules which are abundant in our body and also the brain, and tries to get information of the speed of water molecule movement. So you can imagine it in a way that if you look at a glass of water and you have a lot of water molecules in that glass of water, they can freely move around. That's called random Brownian motion. And as soon as I introduce a barrier into this liquid, for example, by adding, I don't know, a spoon, there are certain parts of this glass of water where the water cannot move freely anymore, but where it is restricted in its movement.
And the spoon would be the traces of the new memory.
Yes, and so if we add new synapses, the water molecules are restricted in their movement, and that means there's less diffusion happening. However, probably we are not imaging these very, very small synaptic changes, but we are imaging a process that is co-occurring with the formation of new synapses. So in the brain we have neurons and we have glial cells. They, for many, many times, they have just been seen as a very passive substrate. They are just like the supporting cells of the tissue. But in the last 15 years, there have been many, many functions that can actually be attributed to these glial cells. And one specific type of these cells is called astrocytes, and they have a massive function in memory formation. So if you experimentally manipulate these cells in animal studies, you can see that memory formation is impaired. And if you have a process of neural activation during memory formation and the formation of new synapses, these glial cells, they swell. So they accumulate water into their cell bodies and increase their volume by a scale of 10, basically. And that probably is the effect that we are seeing when we see changes in the diffusion of water molecules. So an effect that accompanies the formation of new synapses.
And is this effect, like if you are able to locate it and sort of you in the long way, the goal is to interpret this as the storage of a memory trace?
Yes, so that in itself has certain challenges, of course. So what we have been able to see is a very rapid change in diffusivity after memory formation. So when we image 90 minutes after the learning experience or 60 minutes after the learning experience, we can already see this change. And then the question is, how transient is this effect? Is that just induced by acute neural activity and is gone after several hours? Then this is probably not so meaningful for us as a signal. But what we've been able to show so far is that this change in diffusion persists for at least 24 hours. Which makes us think that maybe it's not only the astrocytes that we are imaging, because you would assume that these regulate back to their normal size after 24 hours, especially during sleep. And if you image after 24 hours, there is a period of sleep normally in between. But the question at the moment is, of course, how far can we go? How long can we see these changes? How long lasting are they? And can we maybe make the modeling of our signal that we are acquiring a bit more specific to these actually learning-induced changes?
Okay. Maybe to wrap this up, are there any practical implications that we can take from your research? For example, say if you're a student preparing for an exam?
Yes. So there's actually two roads that this research could go down in the terms of applicability to everyday life. So one you already mentioned, that is education, right? If we know that there are certain ways, and then it would go into the idea of optimizing learning schedules, for example. If we know that there are certain time delays where a repetition is more effective than another time interval, then that could actually inform school and education. If you are repeating a certain kind of material, when should you do that? In what kind of format should you do that? Should the pupils either re-encode that or rehearse that? That is something that we are investigating at the moment, what different time intervals or different forms of these schedules of restudying and rehearsing, so recalling, actually due to the quality of a memory. and to the longevity of a memory, basically. The other avenue that this could go down is the question, if you are relying in the natural case for a relatively long time on your hippocampus, what happens if the hippocampus is dysfunctional? And that is a question that then goes more into the idea of aging, because we know that on the one hand, in pathological conditions such as Alzheimer's, the hippocampus is one of the brain regions that is affected very early in this disease. But also during healthy aging, the hippocampus is one of the systems that shows atrophy as one of the first. And if you think that by using this rehearsal technique to actually make memories rely on the cortex much faster, that could be a compensation strategy if your hippocampus is dysfunctional.
That's super interesting. Since you touched upon aging as well, I mean, I'm not exactly sure about the research behind this, but just from experience, it seems like not everyone is affected by atrophy in the hippocampus in the same way. And I was wondering about the inter-individual differences about these mechanisms that we touched upon here. So, for example, are there people who rely more on their hippocampus and other people that rely more on their neocortex for forming memory?
I think that that is a question that goes much into what strategies do people use to actually memorize content, not even consciously, but maybe even subconsciously. There's people that focus more on, for example, or connect an experience that they make to maybe the verbal system, and this is something that is very cortically grounded, basically. So if you are a person that, if you are memorizing or recalling something, are relying much on like repeating the experience with words in your head, that will probably make it more cortex dependent. Whereas if you are a very visual person, this might actually lead to that you are more prone to store things in the hippocampus, because the hippocampus has this very, very strong connection to the visual system, actually. But then on the other hand, if we talk about vulnerability for hippocampal atrophy in age, that is something that is on the one hand dependent on genetic factors. There's, for example, also different variants of how the blood supply to the hippocampus is formed out that makes a difference in how vulnerable this system is, also to atrophy. But there's also questions of lifestyle. So we know that, for example, people who are physically active also in older age, that they are less affected by hippocampal atrophy, but also brain atrophy in general.
So would you say that everyone has these two systems available, and the specific way in which we use them pretty much depends on how we approach memory problems and how we live and how we use them? So it can be trained or changed?
Yeah, like the plasticity of how these two systems contribute, I think that's a very difficult and unsolved question at the moment. But for sure, these two systems, the hippocampal and the cortical systems, they are hardwired. And everybody uses them probably to a similar extent. So there is, if you look at the natural case, you will always require the hippocampus to encode a stimulus. And that has been shown in many, many people that have hippocampal dysfunction. However, with this, for example, this rehearsal technique, this might actually be a way to, in not so severe cases, push the reliance of a memory on the cortex. And actually, this might even work in hippocampal amnesics, because there are some studies that show that if you repeat a kind of information that is connected to your prior knowledge many, many times, even these people might be able to remember, at least unconsciously in some form. So I think there are limits to this technique, but a certain way of shifting the contribution of these systems is possible.
I'm really curious what's going to come out of this research direction in the future and how patients can benefit from it in real life. But for today, unfortunately, we have to stop the discussion at this point. But, dear listeners, if you're hungry for more answers, don't despair. We're going to continue the conversation in the next episode. While today we focused really on memory and delved into how the hippocampus and the neocortex need to collaborate to form lasting memories, in the next episode we're going to shift our viewpoint a little bit. We'll look more deeply into why sleep is so important for remembering and why we need different sleep phases to properly function. But for now, a warm thank you to Svenja Brodt for generously sharing her knowledge and perspective, and to Simon Faßnacht for co-hosting and steering the conversation with such insightful questions. I'm looking really forward to continuing the conversation. And as always, thanks to all our listeners who tuned in today. You can find links to further information in the show notes. Don't forget to subscribe, share, and tell a friend about signals and synapses. We'll be back soon. Until then, stay curious and keep listening.